pollen grains are generally spherical and measuring about 25 to 50 micrometers in diameter. It has prominent two-layered wall, which is a hard outer layer is called as exine. It is made up of spirochete, which is one of the most resistant organic material. So, spirochete, which is one of the most resistant organic material known. It can withstand high temperature and strong acids and alkali. No enzyme that degrades pollen is so far known. So, pollen grain sign has prominent aperture called joint folds where sporopollen is absent. So, pollen grains are well preserved as fossil because of the presence of sporopollen. The egg sign exhibits a fascinating array of patterns and designs. Okay, now the inner wall of the pollen grain is called intine. It is a thin and continuous layer and it is made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by plasma membrane. The pollen grain is mature. When the pollen grain is mature, it contains two cells, the vegetative cell and generative cell. So the vegetative cell is bigger, has abundant food reserve, and large, irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle-shaped with hidden cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains are shared in this two-cell stage. In the remaining species, the generative cell divides mitotically to give rise to the two male gametes before pollen grains are shared. That means at the 3-6 stage. Okay, so pollen grains of many species cause severe allergies and bronchial affections in some people, often leading to chronic respiratory disorders like asthma, bronchitis, etc. It may be mentioned that Parthenian or carrot grass that came into US containing carrot here. Pollen grains to have pollen grains are rich in nutrients. It has become fashion in recent years to use pollen tablets as a food supplement in Western countries. A large number of pollen products are in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and race horses. Okay, so again, the pollen grains are shaped. Pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. तो अब ये जो pollen grains होते हैं वो कब तक viable रहते हैं मतलब कब तक वो fertilization में part ले सकते हैं तो the period of the period जिसमें pollen grains viable रहते हैं वो highly variable करता है to some extent it depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollens lose viability within 30 minutes of their release, and in some members of roseaceae, leguminous, solanaceae, they maintain viability for months. You may have heard of storing semen, sperms of many animals, including humans, for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen at minus 196 degrees Celsius. Such stored pollen can be used as a pollen banks, similar to seed banks in crop breeding programs. Vein. Now the crystal megasporangium oval and embrace. The gynoecium represents the female reproductive part of the plant. The gynoecium may consist of a single crystal that is monocarpally or may have more than one crystal, multicarpally. When there are more than one, the crystal may be fused together, that is syngalus, or may be free, that is hypogalus. Each crystal has three parts: the stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is a elongated slender part beneath the stigma. The basal bulb part of the crystal is the ovary. Inside the ovary is the ovarian cavity, that is called as the vacuole. The placenta is located inside the ovarian cavity. Okay, so this is the crystal of a flower. It consists of a stigma, style, and ovary. The stigma, it is the landing platform for pollen grains. Style, it is the elongated slender part beneath the stigma, and then the ovary. It is the basal bulb part inside the ovary, which is the basal bulb part, और इसके अंदर जो होता है ओवरी, ओवरी के अंदर होती है ओवेरियन कैविटी जिसे हम वैक्यूल भी कहते हैं. तो जो ओवरी होती है वो इट कन्वर्ट्स इनटू फ्रूट आफ्टर फर्टिलाइजेशन, एंड जो ओव्यूल होता है इट कन्वर्ट इनटू सीड्स आफ्टर फर्टिलाइजेशन. Arising from the placenta are the megasporangia, commonly called as ovule, and the number of ovules in an ovary may be one, that is, for example, in wheat, paddy, and mango, or maybe there are too many like in papaya, watermelon, and orchids. Now, let's learn about the megasporangium, that is ovule. Yeah. Structure of a typical angiosperm ovule. We can see here. Yeah. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with funicle in the region called hilum. Thus, hilum represents the junction between the ovule and the funicle. Each ovule has one or two protective envelope called integuments. Integuments encircle the nucleus except at the tips where a small opening called the macropyle is organized. Opposite the macropyle end is the chalaza, representing the basal part of the ovule. Enclosed within the integuments is a mass of cells called the nucellus cells. Of the nucellus, cells of the nucellus have abundant reserve food materials. Located in the nucellus is the embryo sac or cilium gametocyte. An ovule generally has a single embryo sac formed from a megaspore. And the process of formation of megaspores from the megaspore cell is called as megasporogenesis. Ovules generally differentiate a single megaspore cells in the microbial region. In the microbial region of the nucleus. It is a large cell containing dense cytoplasm and a prominent nucleus. The the megaspore mother cell undergoes meiotic division, and meiosis results in the production of four megaspores. Now, the female gametophyte. In majority of flowering plants, one of the megaspore is functional, while the other three degenerates. Only one is functional and it develops into female gametophyte, that is embryo sac. And this method of embryo sac formation from single megaspore is termed as monosporic development. Okay, the nucleus of the functional megaspore divide mitotically to form two nuclei, which moves to the opposite poles, forming the two nucleate embryo sac. Two more sequential mitotic nuclear divisions result in the formation of the nucleate and later the four nucleate and later the eight nucleate stages of the embryo sac. It is of interest to note that these mitotic divisions are strictly free nuclear. That is, nuclear divisions are not followed immediately by cell wall formation. After the eight nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte or embryo sac. Six of the eight nuclei are surrounded by cell walls and organized into cells. The remaining two nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cells within the embryo sac. Three cells are grouped together at the microphyllate end and constitute the egg apparatus. The egg apparatus in turn consists of two synergids and one egg cell. The synergids have special cellular thickening at the microphyllate called filial apparatus, which play an important role in the guiding to the pollen tube into the large central cell. The large central cell. what कहाँ चली गई मेरी central cell? Okay. The large central cell, as mentioned earlier, has two polar nuclei. Thus, a typical angiosperm embryocyte at maturity, though, eight nucleate and its several cell. Now the pollination. Now the pollination. हॅलो. आता पॉलिनेशन म्हणजे काय? पॉलिनेशनचे मेकॅनिझम काय? ऑब्जेक्टिव्ह काय आहे? So the transfer of pollen grain to the stigma of a pistil is termed as pollination. आता pollination म्हणजे काय? तर पॉलन ग्रेनचा ट्रान्सफर करणे तर कुठे पिस्टलच्या स्टिग्मामध्ये पॉलन ग्रेनचा ट्रान्सफर करणे याला आपण पॉलिनेशन असं म्हणतात. आता वेगवेगळ्या प्रकारचे पॉलिनेशन आहेत ते डिपेंड करतात की सोर्स काय पॉलनच्या. पॉलिनेशनचे 3 टाईप्स पडतात.